Hello, welcome to Dental Board's exam. This is your online source for dental exam review. Okay, so this lecture today is Biochemistry on Glycolysis Lecture 2. So if you haven't seen Lecture 1, make sure you review Lecture 1 on Glycolysis Key Points 1. Okay. All right, so let's discuss the key points of glycolysis. All right, so you have to know if you're going to study biochemistry or take the dental boards exams, you have to know the overall reaction for glycolysis. Okay, so the overall reaction for glycolysis, we have the glucose, and you add 2 ATPs. So you need 2 ATPs in the initial glycolysis steps, okay, plus 2 inorganic phosphate. So the I there on the, okay. So again, as I was mentioning, as I was saying, so you have glucose plus 2 molecules of ATP, okay. So you need to invest 2 molecules of ATP plus 2 inorganic phosphate. So the I here, it stands for inorganic. All right, so inorganic phosphate plus 4 molecules of ADP and 2 molecules of NAD plus. Then your result should get 2 molecules of pyruvate. Okay, so you should note that from 1 molecule of glucose, 4 ATPs are produced. However, 2 ATPs were consumed in the process. So you invested 2 molecules of ATP here, okay, but you produce 2. So your net, okay, so your net ATP produced is only 2 ATP, okay. So glycolysis produces a net of 2 ATPs, okay. The total ATPs produced in glycolysis from 1 glucose of molecule is 4, but the net ATPs is 2. You have to understand that distinction, okay? All right, so let's move on. All right, so you have to know that the 1 molecule of glucose has 6 carbon molecules, and the 6 carbon molecules is divided into 3 carbon sugars, okay. And the 3 carbon sugars are the 1,3-bisphoglycerate, and the other one is phosphoenolpyruvate. So remember these 2 sugars, okay. So 1 carbon of glucose, 1 molecule of glucose has 6 carbon molecules, and this is divided into 2 3 carbon sugars, okay. All right, so let's move on. So some of the important points. What are the important points to remember here? Okay, so you have to remember that the first stage of glycolysis traps glucose within the cell by phosphorylation, and eventually the conversion to fructose 1,6-bisphosphate. Okay, so remember that point. And the second stage of glycolysis forms 2 trios, meaning 2 3 carbon molecules, okay. And this comes from the fructose 1,6-bisphosphate. And then in the final stage, that's when pyruvate, which is a 3 carbon molecule, is formed. All right, but remember, glycolysis produces 2 molecules of pyruvate. All right, and then substrate level phosphorylation, converting ADP to ATP, occurs at 2 points. Okay, So substrate level phosphorylation, which converts ADP into ATP, occurs at 2 points, and we're going to review that. Okay, all right. So 2 points of substrate level phosphorylation is, 1 point is the conversion of 1,3-bis-4 glycerate into 3-phosphoglycerate. That's 1 point. So okay, so this here produces ATP, and then the other stage, the other one is phosphoenolpyruvate to pyruvate. All right, that also produces ATP. All right, so this is a quick review of key important points that you have to remember if you're studying biochemistry or the glycolytic pathways. All right. Okay, so substrate level phosphorylation. All right. So substrate level phosphorylation refers to phosphorylation of adenosine diphosphate, okay, to form ATP, adenosine triphosphate. All right, and you know the ATP is the energy currency of the body, of the cells. So that is what substrate level phosphorylation means. It means you're adding a phosphate group to ADP to form ATP. Okay, and substrate level phosphorylation is independent of electron transport. All right, so you do not need electron transport to have substrate level phosphorylation. Okay, so you have to keep that in mind. And the phosphorylation of ADP is coupled to the dehydration of the substrate 1,3-bis-4 glycerate or phosphoenolpyruvate. Remember this. So the dehydration of these two molecules forms ATP. Okay. So make sure you review. Okay. Make sure you review these points. You have to know substrate level phosphorylation if you're going to be taking a biochemistry exams. All right, and this is the flow, okay, of the glycolytic pathway. Okay. All right, so you can see here we have a glucose molecule, which is then undergoes enzymatic reaction with hexokinase. Okay, so hexokinase takes this ATP, okay, and adds the phosphate to the glucose molecule. So it adds the phosphate here. Okay, so it adds it here. That's a 6 carbon. That's a 60th carbon. So you're adding phosphate to the 60th carbon. Yeah, so you get glucose 6 phosphate. Okay, and these traps. So these traps, when you form the glucose 6 phosphate, you are trapped. This cannot move outside the cell. So now the glucose, which is able to cross the membranes, okay, is now trapped when you add a phosphate, when you form glucose 6 phosphate. So it's now trapped inside the cell. And once it's trapped inside the cell, now you have the phosphoglucose isomerase, okay, which is going to remember, isomerase converts one sugar to another by rearranging the molecules. Okay, so then you form fructose 6 phosphate. Remember the difference between glucose and fructose in terms of the structure is both of them are 6 carbon sugars. However, glucose has a 6-member ring. However, fructose has a 5-member ring. Okay, and so for you to get the fructose 1,6-bisphosphate, then you need phosphofructokinase. Okay, and so which enzymes which steps need ATP? So this step here needs ATP molecule, and phosphofructokinase here, this step needs ATP also. Okay, So that means you are using 2 molecules of ATP. And this is the overall reaction until you produce pyruvate. So you can review that these steps here. Okay. Now you have to keep in mind a step which is the steps which are the steps you need to remember, the steps which are converting, which are producing ATP, and the steps which are consuming ATP. Okay, so this step here is producing ATP. Okay, another step which is producing ATP is here. Okay, so these 2 steps which are producing ATPs are substrate level phosphorylation, the one you just reviewed. Okay, so you have to remember that. Okay, quick, let's go through. Okay, so pyruvate, this is a molecule you have to understand if you're studying glycolysis. So under anaerobic conditions, pyruvate is converted to lactate or ethanol depending on the organism. Okay, so it's converted to lactate or ethanol. So both lactate and ethanol are both they are 3 carbon molecules. Okay, under aerobic condition, pyruvate is converted to acetyl CoA. Keep that in mind. Okay, all right. So the metabolic pathway followed by pyruvate is usually dependent upon the metabolic state of the cell. Remember that. So if you have oxygen, then you're going to convert the pyruvate into acetyl-CoA. If you don't have oxygen, then this is anaerobic conditions. You're going to, if it's in humans, you're going to produce lactate. If it's in yeast, as we reviewed in the previous lecture, then you're going to produce ethanol. All right. So in yeast and other microorganisms, pyruvate is converted to ethanol, as I just mentioned. Okay, and so the first pyruvate is decarboxylated. Okay, so first is pyruvate is decarboxylated. So here is pyruvate decarboxylation. So you're removing 1 carbon. Okay, so here you're removing 1 carbon and you're producing carbon dioxide. Okay, so remember which step is producing carbon dioxide in glycolysis. Okay, all right, and then this forms an aldehyde. Okay, so here is the aldehyde, and you can tell it's an aldehyde because it has this COH group here. All right, with a methyl group. So this is acetaldehyde. Okay, so pyruvate is decarboxylated to form acetaldehyde, and then this step which consumes NADH. Remember that step. Acetaldehyde, all right, is needed to convert. It needs NADH to form ethanol, and you can see that now this ethanol has 3 carbons. All right. So the first reaction usually is catalyzed by the enzyme pyruvate decarboxylase. Remember that enzyme. Okay. So pyruvate decarboxylase removes 1 carbon from pyruvate to form acetaldehyde. And the second reaction is catalyzed by the enzyme alcohol dehydrogenase. Okay. And usually these steps, alcohol dehydrogenase, when you drink alcohol, usually you have alcohol dehydrogenase and then you have aldehyde dehydrogenase. Okay, so it's like almost opposite when you're breaking down alcohol. But remember these 2 enzymes, okay? Pyruvate decarboxylase and alcohol dehydrogenase. Okay, all right. So moving on. Okay, so the conversion of glucose. So now we're discussing alcoholic fermentation. Okay. So if you're understanding glycolysis, you have to understand also fermentation. So the conversion of the fermentation is refers to the conversion of glucose to ethanol. Okay. And the conversion of glucose to ethanol does not involve any oxidation, since the NADA plus is generated in the alcohol dehydrogenase reaction is consumed in the oxidation of glyceraldehyde 3-phosphate in glycolysis. Okay, so you have to make sure that you remember that the conversion of glucose to ethanol does not involve any net oxidation. All right. Okay, so make sure you remember this reaction here. So fermentation, you have glucose plus 2 inorganic phosphate plus 2 ADPs, okay, to produce 2 molecules of ethanol plus carbon dioxide plus 2 ATPs and water. All right, key reaction to remember. Okay. All right, so pyruvate, we have discussed pyruvate before, okay, a few minutes ago, and now you have to understand that in high organisms, pyruvate is converted to lactate. So in humans, if you are running and you're getting those muscle cramps, you know that you are producing lactate. Okay. So in high organisms, pyruvate is converted to lactate when the amount of oxygen is limiting, meaning if you don't have enough oxygen, all right, that's when you start producing that lactic acid. All right. Now, this often occurs in muscles during periods of strenuous activity, and the enzyme carrying out this reaction is lactate dehydrogenase, LDH, lactate dehydrogenase. So usually in people who have they've been running, okay, usually their lactic acid is elevated if you're going to measure it. Okay, if somebody is in trauma and the muscles are traumatized and everything, they're going to have some high levels of lactate. If somebody is in trauma and they're not breathing well or receiving enough oxygen, okay, remember there's limiting oxygen. You're going to also their LDH levels is going to be elevated because they're going to be producing lactic acid. Okay, so lack of oxygen in humans can lead to lactic acidosis. Okay, all right, and this is the reaction that you need to remember. All right, remember pyruvate, okay, plus NADH with the assistance of lactate dehydrogenase. Okay, remember this is an enzyme, so it's catalyzing, speeding up the reaction. It's going to produce, okay, lactate plus NAD plus. All right. All right. So let's answer this question. Very simple from what I just mentioned. So under anaerobic condition in the muscles, pyruvate is converted into ethanol. True or false? Right? So is this true? Is this false? So one, you have to understand we are talking about under anaerobic conditions in muscles. Okay, so we're going to assume that this is a human or a mammal. Okay, so yes, we're going to here we're going to produce lactate. Okay, as I just mentioned. So you don't produce ethanol, because ethanol, this is a reaction which usually occurs in yeast. Okay, so in this case the answer is false. Okay, it's supposed to be lactate, not ethanol. All right, how about this question? What's the answer? Under aerobic condition, pyruvate is converted to and the aerobic, okay, not anaerobic. So make sure you pay attention to the words. Is it converted to ethanol? Is it converted to citric acid, lactate, or acetyl-CoA? So here acetyl-CoA is already checked, okay, so here's the answer. Okay, so acetyl-CoA is what is produced, and aerobic, and enough oxygen supply. Okay, so this is the end of the review. Make sure you subscribe, like, and share to receive notification on the new lectures. Okay, all right, good luck, study hard, and make sure you stay disciplined to pass your exams. Good luck.